Hi, everyone. I'm Emily Kim, and I'm a rising senior at Troy High.
And I'm Riya. I'm a rising senior at Northville High School.
And our presentation is called Testing Testes. So during the summer, we worked under. During the summer, we worked under Dr. Pilsner with our mentor, Emily, and we tested mice samples that had something called an mTOR complex 2 knockout. And so we were looking to see if there was a correlation between their age and mitochondrial and ribosomal DNA copy numbers. So a little bit of background before we get started. Spermatogenesis is basically the differentiation of germ cells to spermatozoa. And so in order for this to happen, we need something called a Sertoli cell. And these are somatic cells located in the testes, and they maintain the environment in the seminiferous tubules so that these germ cells can differentiate. In between these Sertoli cells is something called the BTB. This is a blood testes barrier that is formed by tight junctions, and they protect the sperm from any toxins and the immune system. And so the reason why it protects it from the immune system is that in the body, sperm are actually considered foreign. And so if it were to come in contact with the blood, the immune system might have attacked.
So here's some background on mTOR. mTOR is a mechanistic or mammalian target of rapamycin. It's a protein that regulates cell functions like growth, proliferation, and survival. So there are two complexes, mTOR complex 1 and mTOR complex 2, that are activated and inhibited by cellular stress. So mTOR 1 loosens the blood testes barrier while mTOR 2 tightens it. So it works kind of like a seesaw. So when mTOR 2 is turned off, mTOR 1 prevails. So we focus specifically on mTOR 2 knockouts, which removes the regulator protein Richter that binds to the mTOR 2 complex. When you loosen the blood testes barrier, it exposes the sperm to the immune system and other toxicants. So we compared that to our wild type, which is a control that still has mTOR complex 2.
And oftentimes, like with a loose BTB, as people age, it becomes looser. And so that's why we're looking at the mitochondrial DNA copy numbers. So the machine that we worked on is called the dPCR. So this is a digital polymerase chain reaction. And so what PCR does is it amplifies the DNA sample that we have in order for us to gain data on it and analyze it. And so we wanted to compare this to actually what's called a qPCR. So the dPCR is digital while the qPCR is quantitative. And the difference between these is that a qPCR uses one well, whereas a dPCR could use up to like maybe 1,000 wells. And so there's a smaller amount of DNA in each well. And so that leads to higher sensitivity and increased precision.
So we use 24 well plates, and each well is filled with a supermaster mix in DNA. Our supermaster mix contains different ingredients, and we changed our restriction enzymes based off our ribosomal and mitochondrial DNA. So we had to change the restriction enzymes to see which would cut it better. We also had an incubation period of 1 hour to allow the restriction enzymes to cut as well. We have cycling parameters, which change based on what works best for our assay. So an example of how we changed it is we decreased our temperature from 60 to 55 degrees Celsius to allow our probe more time to bind with our primer. If you look to the right, our data shows a mice sample, which is on the top, and a human sample, which is on the bottom. This 1D scatter plot, an ideal 1D scatter plot would have good separation between the positive reads, which are the blue bands that you see above the red threshold line, and the negative reads, which are below the red threshold line and are gray. So we also want there to be less snowfall, which are the dots that lie between the negative and positive reads, which is why the human DNA is better optimized for our assay, which is on the bottom.
So during our project, we focused on something called copy number. And this is calculated using two things, APOB and 28S. So APOB is a single gene copy number. And so that means that for each genome in our sample, we'll have one APOB. The difference between APOB and 28S is that 28S is repeated throughout that sequence. And so if you look on this, we have like a line and then there's rectangles. So each rectangle could represent like a copy of 28S, while the whole line represents just one genome. And so to calculate that copy number, we would just divide the 28S number that we got from our machine by the APOB, and that will give us our 28S copy number. So 28S is a sequence in ribosomal DNA. And so ribosomal DNA is composed of those repeating sequences within nuclear DNA, and it codes for subunits for the ribosome. So shown here, we have the large subunit, which is also called 60S, and the small subunit, which is also called 35S. And so the repeating sequence that we're specifically studying is called 28S, which is located in that large subunit. And it's found on chromosomes 12, 16, and 18 in mice. So the reason why we're actually observing 28S is because mTOR induces our DNA amplification. And so that was found in a study that was looking at budding yeast. And so we also saw that there was a correlation between cancer cells and those 28S copy numbers. And the study found that with gastric tumors, it had a higher number of 28S copy number compared to that of adjacent normal tissue cells. So the question we were asking was if there actually is a relationship between mTOR complex 2 knockout and that 28S copy number. And so our hypothesis is that when it is knocked out and BTB loosens, we expect that there would be a higher 28S copy number just because like as you age, your BTB would loosen. And so then a higher copy number is more related to like cancer cells and then sometimes like with mTOR. So here are the results when we analyzed the DNA on mice that had the mTOR complex 2 knockout. And so as you can see here in this box plot, we studied mice that were ages 8 weeks and 1 year. So with 8-week-old mice, they are past puberty, and so they're actually able to produce sperm. But unfortunately, if you look at our p-value in the bottom left-hand corner, it was relatively high. So we can conclude that our data wasn't statistically significant. And this could have possibly been due to our low sample size, but that's the result that we have for now.
So our second study looked at mitochondrial DNA, which is a plasmid or disc-shaped piece of DNA or a circular genome that's found in the mitochondria. Typically, we expect mitochondrial DNA to have a high mutation rate, but that's in the major arc. We're focusing on the minor arc that has lower deletions and mutation rates. So mitochondrial DNA copy numbers is the measure of the amount of mitochondrial genomes per cell. Abnormal counts of mitochondrial DNA have been associated with other diseases like cancer. So we're observing this because usually mitochondrial and mtDNA are maternally inherited. So men with high copy numbers are associated with higher risks of infertility, lower semen quality, and lower probability of pregnancy. Mitochondrial DNA copy numbers has also been associated with a variety of age-related diseases. So we're trying to see if there's a relationship between mTOR2 knockouts and mitochondrial DNA CN. So our hypothesis is that when mTOR2 is knocked out and the blood-testis barrier loosens, that mtDNA copy numbers are higher with an increase in age. So our results, we used a Wilcox and rank sum test, which is a non-parametric t-test that compares the medians of each group because our data is not normally distributed. So if you look at our p-values, we had a 0.56 p-value for the 8 weeks, which is not statistically significant, but you can see that there is a slight increase in the copy numbers for the knockouts compared to the wild type. But for the 1 year sample, we have 0.08 p-value, which is borderline statistically significant. And you can see that there is an increase in the mitochondrial DNA copy numbers for the knockout compared to the wild type. So from our preliminary research, our data suggests that with the mTOR2 knockout and an increase in age, mitochondrial DNA copy numbers tend to be higher.
And so for our conclusion, we noticed that there was a trend with mitochondrial DNA copy numbers. And so in the future, this could potentially be a biomarker for male infertility. And then future research. So there are some things that we didn't get to touch on just because we had a limited amount of time. So if there was more time, we would want to test mice with other ribosomal DNA sequences. So although we didn't find a correlation between 28S and their age, we could look at other sequences, including 18S and 5S. We also wanted to look at different ages for the mitochondrial DNA copy number. So the samples that we tested were the oldest and youngest of the mice, but we also wanted to look at mice that were ages 22 weeks and 12 weeks old. And then finally, we only looked at mTOR complex 2 knockout. And so we wanted to look at mTOR complex 1 knockout to see what would happen if the BTB was tighter.
So we would like to thank Dr. Pilsner, our mentor, Emily, Amos and Drew for their guidance and patience, and we really enjoyed working under you this summer. Dr. Suvorov for the background on mTOR and for the samples we used for our study. Dr. Moore, Dr. Alvaro, Alyssa, and the DTC program for providing us this opportunity.